All right, well, I've taken this, the genomic imprinting. There's two examples of this. We have one example in mice and then one example in humans, and I wanted to compare and contrast them in a sense, but still give you an understanding of what genomic imprinting is. Again, we'll explore these four rules of Mendelian inheritance and how genomic imprinting actually alters this expectation that you have under independent assortment. The genes are not really altered in the sense that mutations, we're not introducing mutations again. Remember, epigenetics is just modifications to the DNA, either the specific genes or the chromosomes, such that expression is altered in some shape or form. Now, genomic imprinting occurs when a segment of DNA is specifically marked. And in this, we're actually talking about methylation, or the addition of methyl groups to regions on the, or to the histones, and also into to the DNA. It can't actually be in the DNA. The effect is maintained throughout the life of the organism, inheriting that marked DNA. Now, this type of inheritance is commonly referred to as monoallelic expression, and that's because either the maternally inherited or the paternally inherited allele is the only one that's being expressed. So, therefore, mono being a single allele expression. Now, in mice, imprinting results in an expression pattern of the paternal being transcribed, the paternal allele being transcribed, whereas the maternal allele is typically not transcribed, and no protein is being made from that maternal allele. And a good example of this is seen with the IGF-2 gene, which encodes a growth hormone, and that's an insulin-like growth factor. If you have functional IGF-2 genes, or a gene, then you will develop into an average-sized mouse. Yes, you get to pretend like you're a mouse. If you have a mutant or non-functional IGF-2, this is typically a loss of function allele, and you tend to be dwarf in terms of size. Now, let's look at how this affects our crosses. So we're starting out here with our mother's genotype, and this mother is, and the father, both are true breeders. The mother is homozygous for two non-functioning IGF-2 alleles. The father has the functioning IGF-2 alleles. Now, in this type of inheritance, what's actually is imprinted is the pattern that's seen in the father. So in this case, the father's allele is actually going to be the one that's expressed, whether it is mutant or not. So on this cross, we see that we have a functioning IGF-2, and it's coming from the father. So in our offspring, we're going to be heterozygous. However, this IGF-2 minus allele that's coming from the mother is actually marked so it cannot be expressed. So it's silent, if you will. And actually that's gene silencing is accomplished through methylation process. So all of the offspring from this mating are going to be normal in size because the normal allele is actually being expressed. Now, if we reverse this and we give the father both of the non-functioning alleles, and again with true breeders, and the mother actually has the functioning alleles, what we'll see still is that the father's allele is the one that's being expressed. So in this situation, because that's the mutant allele, or produces the mutant phenotype, all of the offspring are going to be dwarf, because the father's allele is the one that's expressed in the offspring, whereas, and in this case it happens to be non-functional, whereas the mother's allele, which is functional, is not being expressed. So it's silent. Now, let's think about this in terms of somatic cells and also germline cells, which would include the sperm and the eggs. So here we have an egg that has the mother's IGF2 gene that is silenced. In this case, the mother's mother's IGF gene is actually a mutant form. It's fertilized by the males, which has the functioning IGF2. That one is the one that's actually going to be transcribed in the somatic cells of the mouse. So if the IGF2 functioning allele inherited from the father is expressed, then you're going to have normal mice develop, or normal types of mice. Now, that because we still have meiosis ongoing, the possibility for the eggs in terms of what alleles they're carrying is still going to be half of them are going to be IGF2 functional, and then the other half are going to have IGF2 minus present. However, all of these alleles, whether they're functioning or non-functioning, are going to be silenced in the eggs. So that's the pattern for the females. Here, in this, it's the same idea. In the males, the somatic cells are going to have the IGF2 functioning. The non-functional one is actually silenced because it was inherited from the mother. And then in the offspring of these mice that are heterozygous for the mutant and the non-mutant allele, they're going to have half of their sperm that have the functioning IGF2 and half that have the non-functioning IGF2. However, all of these alleles can potentially be transcribed after they join up with an egg and produce an embryo. So half of the offspring would result in a dwarf phenotype, and the other half of the offspring would result in a normal phenotype. Sorry, skipped a slide there. Okay, so as a result of this, genomic imprinting is permanent in terms of the somatic cells of an animal. But what you can do is that you can actually alter the marking of these alleles as you move from generation to generation, i.e. in the germline cells. That's where you get a change. So it can involve a single gene, a part or all of the chromosome, and in some cases it can be all the chromosomes from one parent. So these types of patterns are gene specific, but then they're also specific to different species and how their inheritance patterns occur, how genomic imprinting occurs in them. So you got to know a lot about this to be able to predict it. So they found it in insects, in mammals, and flowering plants. And I would hazard a guess that the more research is done, the more patterns of genomic imprinting will be identified. And even the possibility that XN activation is linked to genomic imprinting. So let's think about how this marking occurs. It's primarily driven by the process of DNA methylation. This is the marking process, and by this I mean you're marking the gene. So at this level, at the molecular level, you have an imprinting control region that's going to be located near that imprinted gene. And then you get this pattern. This is always a pattern. It's either methylated in the oocyte or in the sperm. Typically not in both, because then you're shutting off both copies of that gene. And most of the time, this causes inhibition of transcription. So let's look at this, what we have here. In this case, in this situation, here we've got the paternal chromosome is the one that's methylated. So in somatic cells, that pattern is going to be maintained. This is referred to as maintenance methylation in all the somatic cells as you go through successive cell divisions to build an organism. Now, in female cells, if we have our maternal chromosome, this methylation pattern that's seen in the somatic cells is going to get erased. And you can essentially have set up your eggs with a non-methylation pattern. Now, this is different from what we just talked about with the mice, but yeah, because it's actually the mother's gene, the IGF gene, that's actually methylated and untranscribed. But what I'm trying to demonstrate here is that this pattern involves either maintenance methylation in somatic cells, erasure, and then either no methylation or de novo methylation, depending on where the methylation patterns are, in what sex, male or female, methylation patterns are laid down. So in this example, we only see methylation in the male gamete cells. So here we get, again, we get this erasure of the methylation in the germline cells, and then de novo methylation of both chromosomes to begin the formation of the gametes produced in this case. So to be able to predict the phenotype of an individual, you have to know the pattern of genomic imprinting for a specific gene. And then you have to know something about that gene, whether it's a mutant version or a non-mutant version.